Hello students, welcome to Read MedPrep Academy channel. Today in Asexual Reproduction in Plants Part 3, we are going to discuss about the artificial methods of vegetative reproduction in plants. A number of methods are used in agriculture and horticulture to propagate plants from their parts. Such methods are said to be artificial propagation. There are two types of artificial methods of vegetative reproduction. One is conventional methods and two is modern methods. Some of these artificial propagation methods have been used by man for a long time and are called conventional methods. Nowadays, technology is being used for propagation to produce large number of plants in short period of time. Such methods are said to be modern methods. Now let us discuss the conventional methods. What are the types? Cutting, grafting and layering. What is cutting? It is the method of producing a new plant by cutting the plant parts such as root, stem and leaf from the parent plant. The cut part is placed in a suitable medium for growth. It produces roots and grows into a new plant. Depending upon the part used, it is called root cutting, stem cutting and leaf cutting. Root cuttings are pieces of roots that form new shoots. These are often made in winter or early spring before plants begin growing. Roots should be the thickness of a thick wire. They are cut in 1 to 2 inch length and placed on a moist rooting medium. Cover and keep moist and they should not be made wet. Example, malice. Here you can see the cutting method of root. A root is cut and is placed in another medium where a new shoot grows and forms a new plant. What is stem cutting? A piece of a plant stem or branch including at least one node is used in propagation. Stem cutting is the widely used method of propagation. Examples, hibiscus, bougainvillea and moringa. So what is done in stem cutting? The stem cutting from a healthy disease-free plants, preferably from the upper part of the plant, is taken. Cutting should generally consist of the current or the past season's growth. And all the flowers and the flower buds are removed from the stem cutting and the stem is placed in another pot or place in the soil and it grows into a new clone or plant. What is leaf cutting? A cutting consists of a leaf instead of a root or a shoot commonly used in propagating a plant. Examples, begonia, bryophyllum. What is leaf bud cutting? This type of cutting consists of a leaf blade, a petiole and a short piece of the stem with an attached axillary bud. This is placed and a new plant grows. Here you can see in this image the leaves are placed and new plants are growing from the axillary buds, forming new roots and shoots. Now what is grafting? In this, parts of two different plants are joined together so that they grow in one plant. Of the two plants, the plant which is in contact with the soil is called the stalk and the plant used for grafting is called the scion. Examples are citrus, mango and apple. There are different types of grafting based on the method of uniting the scion and the stalk. The different types of grafting are bud grafting, approach grafting, tongue grafting, crown grafting and wedge grafting. Let us discuss one by one. In bud grafting, a T-shaped incision is made in the stalk and the bark is lifted. The scion bud with little wood is placed in the incision beneath the bark and properly bandaged with a tape. Here you can see the stalk is taken and a T-shaped incision is made and the bark is lifted and the bud from the scion is placed inside and it is bandaged and this grows into a plant. What is approach grafting? In this method, both the scion and the stalk remain rooted. The stalk is grown in a pot and brought close to the scion. Both of them should have the same thickness. A small slice is cut from both and the cut surfaces are brought near and tied together and held by a tape. After 1 to 4 weeks, the tip of the stalk and the base of the scion are cut off and detached and grown in a separate pot. Here you can see the stalk being brought near the scion and the sides are cut and attached together and they are bandaged. What is tongue grafting? A scion and stalk having the same thickness is cut obliquely and the scion is fit into the stalk and bound with a tape. Here you can see the stalk which is rooted and the scion is brought and is attached appropriately and bandaged. What is crown grafting? When the stalk is large in size, scions are cut into wedge shape and are inserted into the slits or clefts of the stalk and are fixed in position using the graft wax. Here you can see the stalk where the incisions are made and open up, slits are made, and on this the scion is brought and fixed to the stalk. What is wedge grafting? In this method, a slit is made in the stalk or the bark is cut. A twig of scion is inserted and tightly bound so that the cambium of the two is joined. Here you can see the wedge grafting where the scion tip is made into a wedge and the stalk is also cut appropriately and the scion is fixed into the stalk and is bandaged. Now let us move on to layering. In this method, the stem of a parent plant is allowed to develop roots while still in contact. When the root develops, the rooted part is cut and planted to grow as a new plant. Examples, Ixora and Jasminum. There are two types of layering, mound layering and air layering. What is this mound layering? In this method, the plants having flexible branches are used. The lower branch with the leaves is bent to the ground and part of the stem is buried into the soil and the tip of the branch is exposed above the soil. After the roots emerge from the part of the stem buried in the soil, a cut is made in the parent plant so that the buried part grows into a new plant. Here you can see the flexible branch is buried under the soil and a mound of soil is placed, or sometimes a weight in the form of a stone can be placed on the mound so that the branch does not come out and a new roots develop in this branch and this branch is cut off from the parent plant and this grows into a new plant. What is air layering? In this method, the stem is girdled at the nodal region and hormones are applied to this region which promotes rooting. This portion is covered with damp or moist soil using a polythene sheet. Roots emerge in these branches after 2 to 4 months. Such branches are removed from the parent plant and grown in a separate pot or ground. Here you can see on the left side, a bark tissue is removed in a branch of a plant and the moist soil along with the hormones is applied around the cut part and a polythene is tied around to retain the moisture, and once the roots start developing, this branch is separated from the parent plant and is grown separately. What are the advantages of conventional methods? The plants produced are genetically uniform. Many plants can be produced quickly by this method. Some plants produce little or no seeds. In others, the seeds produced do not germinate. In such cases, plants can be produced in a short period of time by this method. Some plants can be propagated more economically by vegetative propagation. Example, potato, Solanum tuberosum. Two different plants with desirable characters such as disease resistance and high yield can be grafted and grown as a new plant with the same desirable characters. What are the disadvantages of conventional methods? Use of virus-infected plants as parents produce viral-infected new plants. Vegetative structures used for propagation are bulky and so they are difficult to handle and store. Now, what are the modern methods of vegetative reproduction? Here we are going to discuss about plant tissue culture. Examples of plant tissue culture are carrot and tobacco. What is the definition of tissue culture? It is an in vitro cultivation of plant cell or tissue under aseptic or controlled environmental conditions in liquid or on semi-solid, well-defined nutrient medium for the production of primary and secondary metabolites or to regenerate a new plant. What are the types of plant cultures? Callus culture, cell suspension culture, anther culture, ovule culture, embryo culture, protoplast culture and micropropagation. Let us see one by one. Here you can see the plantlets of sundew in the culture media in a petri dish. Here you can see the plantlets in a laboratory in the bottles containing the culture media. Now the first is the callus tissue culture of carrot plant. The tap root, which is a carrot, is taken and is washed. It is broken into 3 or 4 pieces and a cork borer is inserted and a core is made. The core is cut and is placed in a petri dish. The discs is placed in an initiation medium. Callus forms in 4 to 6 weeks. Transfer to the root development medium. Root and shoot development occurs in 4 to 6 weeks. And this is transferred to the soil. And this develops into new plants. And these new plants will have development of tap roots which contain the carrot vegetable. Now what is the cell suspension culture? Here the callus that is formed is taken in a liquid medium and is agitated by a shaker so that the cells separate and they form a cell suspension solution. And this is filtered and this filtration is washed so that the individual cells are separated and observed under the hemocytometer. The cell suspension medium, the cells are taken and are plated on a culture medium which contains nutrients and these develop into cell aggregates. And these form embryoids, develop into small plantlets and these plantlets are planted in the soil. Now, what is anther culture? Anther is taken from the flowers and they are cultured in a nutrient medium. This anther can develop into an embryoid and form a haploid plantlet, or this anther can develop into a callus and may form a haploid plantlet. Now what is ovule culture? Ovule culture is an elegant experimental system by which the ovules are aseptically isolated from the ovary and are grown aseptically on chemically defined nutrient medium under controlled conditions. Here you can see image A where the ovules are separated from the ovary and these ovules in the image B are grown in a nutrient medium which forms the plantlets. What is embryo culture? We must know that the seed has a seed coat and it has a cotyledon which stores the food, and they have an embryo which is formed after fertilization. This embryo is formed as a zygote from the fusion of the male and the female gamete. This embryo is cultured in a nutrient medium and these embryo develop into small plantlets. You can see in the image on the right side. What are the advantages of the embryo culture? Using embryo culture, seed dormancy can be overcome. Embryo rescue, which can circumvent post-zygotic barriers. To raise the rare hybrids by rescuing embryo from incompatible crosses is the most popular application. The production of embryos from somatic or non-germ cells usually involves a callus intermediate stage which can result in variation among the seedlings. What is the protoplast culture? Here, a young plant is taken. From the young plant, the leaf is taken and it is sterilized and the epidermis is peeled. The epidermis consists of the epidermal cells and these cells are plasmolyzed and the cells are put in an enzyme solution and the cell wall digestion occurs and results in the formation of a protoplast. And the cell debris is removed and the protoplast is washed and is plated in a nutrient medium. And this protoplast develops a cell wall and forms clumps of cells, and these clumps of cells form the callus. The callus slowly develops or differentiates into different organ tissues and forms plantlets, and each plantlet is planted in the individual pots or soil. Now what is this micropropagation? In stage 1, the explants are taken which are soft wood shoots, and these shoots are established in culture 2 to 3 months. In stage 2, the shoot multiplication is done, where in the cycle 1 there is a development of small cluster of shoots. In cycle 2 there is small clumps of shoots increase in 2 months, and in cycle 3 normal increase in 2 months of shoots occur. In stage 3 the rooting is done. Rooting is done in vitro in a culture which takes about 1 month, or ex vitro rooted directly in the soil 1 to 2 months. And these plants established in the individual cells takes 3 to 4 weeks and are placed in mist. And the total growth period in cells is about 3 months. And the individual plantlets that are formed are grown in individual pots or in the soil when they are grown much. Now, what are the advantages and disadvantages of plant tissue culture? The advantages: a large number of plants can be produced in a short period of time. The technique provides the opportunity to control growth conditions and to synchronize the processes of growth and development, making it possible to obtain plants with optimal characteristics. New genes can be introduced into the plants, which boost crop yield or confer resistance to pests and infections. What are the disadvantages? All the plants that are produced have the same genetic material, so they are equally vulnerable to environmental factors, infections and pests. No new combination of traits are formed, which only results from meiotic division can be produced. So the genetic diversity of the plants is reduced, and some alleles can be irreversibly eliminated from the gene pool. So today, in asexual reproduction in plants part 3, we discuss about the artificial methods of vegetative reproduction in plants, the conventional methods and the modern methods. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.